Hello everyone, this is Joe and thank you very much for having me here. Now I'm serving as Product Development Manager in ACROBiosystems, which is a leading manufacturer of recombinant proteins and other critical reagents to support the development of target therapeutics. I will provide detailed introduction about ACROBiosystems to you later. This talk, I'm going to discuss about the crucial role of CD3 protein in the development of anti-CD3 bispecific antibodies. And I will start with the rapid development of monoclonal antibody industry. In 1997, the approval of rituximab by the US FDA marked the monoclonal antibody-based therapy field entering into the booming stage. Since then, about 100 antibody-based therapeutics have been approved by the regulatory agencies worldwide, and antibody therapeutics have now become one of the mainstays for developing new medicines. The history of development of bispecific antibodies almost followed the footprint of the development of monoclonal antibody therapeutics. A bispecific antibody is a kind of artificial protein that combines to two different antigens or two epitopes of one antigen simultaneously. Bispecific antibodies can play a synergistic role of the two monoclonal antibodies and have advantages over the simple combination. Because of its unique biological mechanism, the bispecific antibody has become an important branch of therapeutic antibody research and development. According to the data source, the number of clinical studies using bispecific antibodies has increased exponentially in the past 14 years. As of September 2019, there are more than 110 bispecific antibodies in clinical trials and about 180 in preclinical development worldwide. And currently, only 3 bispecific antibodies are commercialized, and only 4 products are in phase 3 studies. Most bispecific antibodies in clinical development are in early studies, with 67% in phase 1 and 25% in phase 2. The majority of clinical stage bispecific antibodies are designed to treat cancer, especially solid tumors, breast cancer, and acute myeloid leukemia. Nevertheless, there are some products designed to treat other conditions such as rheumatoid arthritis or autoimmune diseases. The 3 commercialized bispecific antibodies include Removab from Triumph Pharma, that target EpCAM and CD3, Blincyto from Amgen, that target CD19 and CD3, Hemlibra from Roche, that target coagulation factors. 2 out of the 3 launched bispecific antibodies, Removab and Blincyto, target CD3 for cytotoxic effector engagement, which will be discussed in detail later. Removab was first authorized for market by the EMA in 2009 for the treatment of malignant ascites, but was withdrawn in 2017 due to commercial reasons. On the other hand, Blincyto was authorized by FDA in 2014 for treating acute lymphoblastic leukemia, and it has shown impressive clinical results since its launch. Hemlibra is approved by FDA in 2017 to prevent or reduce the frequency of bleeding episodes in adult and pediatric patients with hemophilia A who have developed antibodies called factor VIII inhibitors. With the increase in bispecific antibody development, the number of deals have focused on clinical stage bispecific antibodies, resulting in an increase of 140% in the last 3 years, while the total disclosed deal value decreased by 50%, from $3.2 billion to $1.6 billion. From these deals, 9 were worth more than $100 million. Sanofi and Regeneron's 2014 co-development agreement focused on various antibodies. From these deals, $3.8 billion was spent on oncology, followed by $1 billion on infectious diseases, from a total of $6.8 billion. The global market of bispecific antibodies was worth $0.46 billion in 2018, which was dominated almost equally by Blincyto and Hemlibra. As predicted, the market for Hemlibra will boom in the next few years and grow up to $3.96 billion by 2024. Instead, Blincyto will only have a moderate increase, With the massive sales growth of Hemlibra and the potential approval of new entrants, the bispecific antibody market will surpass $5.43 billion in 2024. It is notable that the strategy in nearly 40% of the developing bispecific antibodies is to deplete the tumor cells by engaging cytotoxic effector T cells using CD3 targeting arms. Here shows a network graph characterizing the target pairs of most bispecific programs in both preclinical and clinical investigations. Each node in the network is one target, and each edge connecting two nodes represents one bispecific program. The circular edges are biparatopic programs. The node size shows the degree of a particular target being paired with other different targets. The color of the edges are marked in black if only one program is available for that particular pair, otherwise in red if more than one are being explored. The popularity of that bispecific program is reflected from the thickness of the red edges. As you can see from the graph, the node size representing CD3 is the largest among all the targets. The number of edges and the thickness of the red edges connecting CD3 and other targets indicate the popularity of this molecule in bispecific antibody development. CD3, also known as cluster of differentiation 3, is a protein complex and T-cell co-receptor that is involved in activating both the cytotoxic T cell and T helper cells. It is composed of 4 distinct chains. In mammals, the complex contains a CD3 gamma chain, a CD3 delta chain, and 2 CD3 epsilon chains. These chains associate with the T cell receptor and the Z chains to generate an activation signal in T lymphocytes. The TCR, Z chain, and the CD3 molecules together constitute the T cell complex. Because CD3 is required for T cell activation, anti-CD3 bispecific antibodies are a promising therapeutic approach for the treatment of cancer. These molecules bind to CD3 in the T cell receptor and tumor-associated antigen on the tumor cell surface. The linking brings the T cells and tumor cells in close proximity and results in TCR clustering at the center of the immune synapse. Following TCR clustering, cytolytic granules move toward the center's supramolecular activation cluster and release perforin and granzymes into the target cells, followed by activation of target cell caspases and apoptosis. Once the target cells undergo cell death, the T cells quickly detach from the dying cells and move to the next target cell. Bispecific antibodies with an Fc region retain Fc-mediated effector functions, such as antibody-dependent cell-mediated cytotoxicity, antibody-dependent cellular phagocytosis, and complement-dependent cytotoxicity. Let's take Blincyto as an example. It is constructed by genetically linking the minimal binding domains of monoclonal antibodies for tumor-associated surface antigens and the T-cell receptor-associated molecule CD3 onto a single polypeptide chain. The minimal binding domain are 2 single-chain variable fragments with binding specificities for CD19 and CD3, and it can be used to treat acute lymphoblastic leukemia. Since CD3 is a popular target in bispecific antibodies development, it should be noted that high-quality CD3 protein plays a crucial role in antibody discovery and subsequent in vitro and in vivo tests. For example, CD3 protein is usually applied in immunization and in vitro screening scenario. In the process of developing anti-CD3 bispecific antibodies by Regeneron, first, they immunized genetically modified mice with the CD3 antigen, and then a pool of high-affinity CD3 specific antibodies were derived from the generated B cells. Next, the generated antibodies were tested for affinity to human and cynomolgus monkey CD3 antigen, in an in vitro binding assay, and one CD3 antibody was selected as a candidate because it was able to bind to both human and cyno CD3 with an EC50 between 1 and 40 nanomolar affinity using FACS. ACROBiosystems has designed and produced a variety of CD3 proteins for the development of anti-CD3 bispecific antibodies. Before introducing the product pipeline of CD3 proteins, I would like to offer you a more detailed introduction of ACROBiosystems. ACROBiosystems is founded in 2010, and we are a leading manufacturer of recombinant proteins and other critical reagents to support the development of target therapeutics. ACROBiosystems is dedicated to accelerating the process of target therapeutic drug development from the starting point of target identification to endpoint of clinical trial. ACROBiosystems has served over 4,500 customers from around 60 countries and reached out to 20,000 end users from pharmaceutical companies and academic institutes. ACRO has established long-term collaboration with big pharma such as Pfizer, Novartis, Johnson & Johnson, and well-known academic institutes like NIH. To provide better service to end users, ACROBiosystems has set up 2 working places at Beijing and Shanghai in China, and 3 offices at Boston, San Jose, and Newark in US. In order to better serve for the bispecific antibody development, ACROBiosystems has produced a variety of CD3 protein products, which will facilitate the discovery and screening of anti-CD3 bispecific antibodies. The features of our CD3 products include stacked mouse verified heterodimer, bioactivity verified by clinical bispecific antibody, batch-to-batch quality consistency, multiple species and different expression tags. The web link below the graph is our CD3 products landing page, and you can visit the website for more detailed information. This slide shows the CD3 heterodimer products from ACROBiosystems. Up until now, we have developed 13 products which are from human, cynomolgus, and mouse, and they are with tags like his tag, human FC tag, and his ABI tag. The products with his ABI tag are in Biotinylated form and are usually applied in ELISA and surface plasma resonance assays. ACROBiosystems are the only reagent vendor which has developed an exclusive collection of pre-labeled and experimentally validated biotinylated CD3 proteins. The products are produced using the his ABI tag technology, which introduces a unique 15 amino acid peptide ABI tag into the recombinant protein during expression vector construction. The single lysine residue in the ABI tag is enzymatically biotinylated by the ACROBiotinylated SPR&BLI. The advantage of this technology is that the biotinylation only happens on the single point lysine residue of ABI tag, and the protein orientation is uniform when immobilized on an avidin-coated surface. In addition to CD3 heterodimer products, ACROBiosystems also provides CD3 proteins. The 3 CD3 products are from human and cynomolgus and are with his and human FC tag. For CD3 we have developed 7 products from human, cynomolgus, and they are with his, human FC, and his ABI tag. In terms of CD3 2 products from human and cynomolgus with his tag are listed in our website. Because the CD3 protein exists mainly as heterodimer on T-cell surface, it is key to recombinantly produce CD3 and CD3 heterodimer that mimic the natural form of these protein complexes. In expression attempts, CD3 and CD3 or CD3 are usually individually transfected into host cells, and ideally the yielded proteins are in heterodimer form. However, it is very difficult to distinguish between CD3 or CD3 heterodimer from their homodimer form. ACROBiosystems has tested lots of constructs of CD3 proteins to screen for a construct that mostly produce heterodimer form. After multiple attempts, we have developed a series of homogeneous CD3 heterodimers with good purity and bioactivity. The 1-to-1 heterodimer ratio was verified by reductive and non-reductive electrophoresis and multiple angle light scattering technology. As shown here, the sample of his tag human CD3 CD3 heterodimer protein was run on SDS page under reducing and non-reducing conditions, and the ratio between CD3E and CD3D in reducing condition was close to 1-to-1 ratio. The molecular weight of this product is around 35 to 43 kilodaltons. That is verified by SecMols. To verify the bioactivity of CD3-ED heterodimer, we conducted ELISA to test the binding ability to well-recognized anti-CD3 monoclonal antibodies. The result shows that immobilized his-tagged human CD3-ED heterodimer protein combined to OKT3 and UCHT1 with a linear range of 0.2 to 6 nanogram per milliliter, which is consistent with published reports. We also did clinical PK assay using in-house developed bispecific T-cell engager in human serum, and the CD3-ED heterodimer protein his-tagged human CD3-ED heterodimer protein is immobilized in the ELISA played well. Then increasing concentrations of bispecific T-cell engager targeting CD3 and bsAb in 10% human serum and biotinylated bsAb were added to the plate well. Detection was performed using HRP conjugated streptavidin, and the result shows that the sensitivity is 4 nanogram per milliliter. To further verify the bioactivity of this CD3 product, we conducted surface plasma resonance experiment. His-tagged human CD3-ED heterodimer protein was captured on CM5 chip via anti-His antibody, and it shows that this heterodimer combined to monoclonal anti-human CD3 antibody OKT3 with an affinity constant of 40 nanomolar as determined in SPR&BLI with BF or T200. In another experiment, bispecific T-cell engager targeting CD3 and bsAb was immobilized on CM5 chip, and the result shows that the bispecific antibody combined to his-tagged human CD3-ED heterodimer protein with an affinity constant of 31.8 nanomolar as determined by BF or T200. These data are all consistent with published literature. The batch-to-batch consistency is a big concern for antigen protein vendors, and ACROBiosystems have conducted a quality control experiment to see the difference among batches. Biotinylated human CD3-ED heterodimer protein from 3 lots are immobilized on streptavidin pre-coated plate, and the CD3 heterodimer combined to monoclonal anti-human CD3 antibody, OKT3, with a linear range of 0.2 to 6 nanogram per milliliter, and batch differences EC50 is below 0.0001 microgram per milliliter. The result shows that the products from different lots have almost the same bioactivity, and the batch-to-batch difference is minimal. Now, to conclude this talk, anti-CD3 bispecific antibody development is a hot area in immunotherapy, and high-quality CD3 protein are important for the successful discovery and screening of the CD3 bispecifics. ACROBiosystems has tested multiple constructs of CD3 heterodimer proteins, and we have finally screened out the best construct that yields homogeneous CD3 heterodimer. Our in-house ELISA and SPR experiments show that the CD3 heterodimer have high bioactivity, which is consistent with published literature. The strict quality control in ACROBiosystems can guarantee the minimal batch-to-batch differences of our products. So, that's all for my part, and I would like to invite you to visit our website for more detailed information if you are interested in CD3 bispecifics development and also other therapeutic targets. Thank you very much.